Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Um, Last week I have talked about the drug targets. So drug can act on the molecular target located in the cell membrane of the cells within the cell themselves. Right. Drug targets are usually the macromolecules, right? And they have bind into the drug, fit and uh, switch on or switch off the drug activity. So most of the macromolecules are the protein in nature, right? So most drugs bind to their target by means of intermolecular attractions or intermolecular bonding. So the study on which the how the drug act or how the drug bind to the target and produce the pharmacological effects is called the pharmacodynamics. So we have talked about various intermolecular interactions which involve in the binding of the drug with the respective macromolecule or drug targets. So these are intermolecular force, intermolecular bonds are the electrostatic or ionic interactions which occur between the groups and opposite charges. Hydrogen bonds, which usually occur in between the electron-rich heteroatom and electron-deficient hydrogen. Hydrogen bonding also involved in this hydrogen bond donors and hydrogen bond acceptor. So the electronegative atom that interact with the hydrogen in a hydrogen bond is called hydrogen bond acceptor. Another intermolecular bonding is the van der Waal interactions, which take place between known molecular region of the molecules and are caused by the transient dipole-dipole interactions. And then the ion dipole and dipole-dipole interactions, these are generally the weak form of the electrostatic interactions. Hydrophobic interactions, which involve the displacement or outer layer of the water molecule which surround the hydrophobic region of the molecule, the resulting increase in generally increasing the entropy contribution to overall binding energy. Polar groups have to be desolvated before intermolecular interactions take place. So, and then we have covered the structure of the protein, right? So there are there are four type of the uh, mm, structure of the protein: primary structure, secondary structure, tertiary structure, and quaternary structure. So the each amino acid bind with another amino acid through the peptide bonds, right? So usually the tertiary protein structures generally shows that how the drug interact with the drug targets through various type of the bonding, various type of intermolecular bonding. So usually in the bonding between the macromolecules and or the drug targets and the drugs are the same as we have seen previously, that ionic bonding, hydrogen bonding, uh, and then the dipole-dipole interactions, which hold the molecule or which hold the drug uh, tightly into the drug target. And then we have seen uh, the the most important one drug target is the enzyme, which so the function of the enzymes are the catalytic function. So enzyme are the protein which act on as the body's catalyst agent that speed up the chemical reaction without being consumed themselves. So enzyme provide the reaction surface and suitable environment. Enzyme bring the reactant together and weaken the bonding in the reactant. Enzyme may participate in the mechanism, and enzyme form the stronger interaction with the transition state than the substrate or the product. So there are the enzyme also. The most important function of the enzyme is the catalytic role, right? So enzyme the drug bind with the respective enzyme through the binding interactions, right? So these binding interactions can be the hydrogen bond interactions or ionic bond interactions. As strong as the drug binds to the drug target, it uh, show the significant activity. For example, if one compound is active and another compound is inactive, it means that the active compound have stronger interaction with the enzyme or any drug target as compared to the compound which is inactive, means it has weakened bonding or no bonding with the specific drug targets. So we have seen that how drug target enzyme and inhibit their action. They can act as a inhibitor, act at the active site of the enzyme. They can be the reversible inhibitor or irreversible inhibitor. The difference is only the at the binding site. If the drug binds through intermolecular interaction, so this type of inhibition will be the enzyme, the reversible inhibition. If the drug bind with the enzyme via covalent bond, then this type of the inhibition is irreversible inhibition. So generally the competitive inhibitor bind to the active site through intermolecular bond, and so their binding is irreversible. Right. So there are many examples of useful drugs that act as a competitive inhibitor. For example, sulfonamide, which act as an antibacterial agent, uh, proton pump inhibitors and nerve agent. They act as an irreversible inhibitor. They bind with the active site of the enzyme through the covalent bonding, right? And then we have seen the some of the inhibitor, which are the allosteric inhibitor that bind to the allosteric binding site, which is different from the active site. They alter the shape of the enzyme, deform the shape of the enzymes, and such that the active site is no longer recognizable by the substrate. Transition state analogs are the enzyme inhibitor, which designed to mimic the transition state of the enzyme catalyzed reaction mechanism. They bind more strongly either with the substrate or with the product. There are some substrate which act as a suicide substrate uh, that act as a substrate for target enzyme, but they convert it into the highly reactive species as a result of enzyme catalyzed reaction mechanism. And then there we have seen some of the medicinal uses of the enzyme inhibitor. These inhibitors are the used against the microorganism, used against the virus, used against the own body enzyme. So these are the examples of the drug act on active site of the enzyme and inhibit the function or deactivate the function of the enzyme. So the most important thing is how strongly the drug or the substrate bind with the active site of the enzyme, right? Which indicate that the compound or the drug is active or the inactive. Now we are moving to the next drug target, which is the receptor. Yeah. So the receptor are the proteins which are by far the most important drug targets in medicine. So the receptor and their chemical messenger are crucial to the communication system of the body. Such communication is nearly essential to the normal working of the body. This is implicated in alignment such as pain, depression, Parkinson's disease, heart failure, So the receptor are identified by the specific neurotransmitter or hormone which activate them. Thus, the receptor activated by the dopamine is called the is dopaminergic receptor. The receptor activated by the acetylcholine is called the cholinergic receptor, and the receptor activated by the adrenaline or non-adrenaline is called the adrenergic receptor. So, receptor is a protein uh, usually embedded in the cell membrane with part of its surround exposed on the outside of the cell. So the protein surface is a complicated shape containing this a hollow, right? Containing the hollow and somewhere within the complicated geography there is a area that has the correct shape to accept the incoming messenger. And so this area is called the binding site and it's analog to the active site of the enzyme. That's how this messenger binds and the message is transferred inside the cell and then the cell will respond. So the target cell containing various receptors specific to different type of the messenger. For example, this is the different receptors, which is for the different type of different various receptors, which is for the different type of the messengers. So these are the different receptors. And for example, cholinergic receptors, which is having the nicotinic muscarinic receptors as well as the nicotinic receptor, and so it has subtype nicotinic receptor M1 to M5 for muscarinic receptor and nicotinic and muscarinic receptor. Right? So it simulates stimulation of the gastric intestinal tract, right? And then the example of antagonist is neuromuscular blockers and this muscle relaxant. So this is the same as for the adrenergic receptors, histamine, um, and then the opioid and opioid-like receptors and these 5-hydroxy tryptamine, right, estrogen, right, which is agonist is contraception, and the example of antagonist that is breast cancer. So these are the different type of the receptors. So this is the binding of the chemical messenger to the protein receptor. So this is the messenger and the specific site on the where the receptor, which is called the binding site. And so there's an induced fit between the messenger and this receptor, and then message is transferred through this message transferred through the receptor inside the cell, right? And then after this will be this. So the main important thing in which we emphasize is the binding, how the drug binds with the receptor. So, for example, this is a neurotransmitter, right? So this is the hypothetical receptor and neurotransmitter. So this is the receptor, which receptor have the different binding site. For example, this hydroxyl group, this is a carboxylate group, which can bind with the a neurotransmitter through the ionic bonding, and this will be bind with the neurotransmitter through hydrogen bonding, right? So this is how this is the neurotransmitter, and this is how this is bind with the receptor. This neurotransmitter bind with, or induce fit into the receptor through the binding interaction. So as much as strong as the binding interactions, so uh, the affinity of or the action of neurotransmitter or action of the drug is directly respond. So this one you can see this one is the hydrophobic interactions, right? With the aromatic ring, hydrophobic interaction. This is having hydroxyl forming the hydrogen bond, And then this one is the carboxylate, which form the ionic bonding with the amino group of the neurotransmitter. So this is how you can see. This is how the neurotransmitter bind with the receptor. As strong as the binding, the response will be simultaneous. So receptors as a drug target. So drug can play an important role by either acting as a replacement messenger or blocking the receptor from receiving their natural messenger. So they can be the drug can mimic the natural messenger or activate the receptor, which is called the agonist, and drug that can block the receptor are called the antagonist. So we will see how the drug interact with their receptor through binding interaction and produce the pharmacological effect. So this is the main purpose of this lecture. So there are first of all the design of agonist. So how the agonist molecule or molecule that mimic the natural messenger and activate the receptor, how that type of the drug, that type of the messenger structure can be, right. So the following message there are the requirements considered to design the drug which is interact with the receptor in such a way that mimic the natural chemical messenger. So the most important thing, the drug must have correct binding groups. Means that binding groups judges that how strong the molecule bind with the receptor, right? So and the second thing is the drug must have these binding groups correctly positioned. So position the position of the drug group should be defined correctly, and the drug must have the right side, the right size for the binding site. So these are the three main uh, features should be in the drug that can help the drug that can help the molecule to strongly bind the receptor. So one by one, we will see the number one is the binding group. So if we can see there's a hypothetical neurotransmitter and possible binding group. So you can see it has a hydroxyl group, it has a aromatic ring, and this is a amino group which have bind with the ionic interaction. This is a hydroxyl group have to bind with the receptor through hydrogen bonding, right? And this is the ionic interaction, right? For example, here you can see this one is this molecule doesn't have the this molecule doesn't have the aromatic ring, which cannot bind with this hydrophobic part of the receptor, right? And doesn't have the hydroxyl group, which can help to bind the with the receptor through hydrogen bonding. So the bomb the so this is very important that the functional group in the um, uh, molecule should be help the molecule to bind with the receptor via these binding modes, right? So you can see here as compared to this molecule, this one doesn't have the hydroxyl group. So this molecule have less activity, less affinity toward the receptor as compared to this one. So this is how the binding groups play a very important role in the binding of the drug with the receptor. Position of the binding group, yeah, the position of the binding group also makes very different. If let's say for example, if the hydroxyl group is here, right, if the hydroxyl group here is here, it will help to form the hydrogen bond easily. But if the hydroxyl group is very far, so this is a positional isomer, this is a positional position of the binding group is on the next carbon as compared to the previous one, right? As compared to the previous one, this is adjacent carbon, but here it is on the next carbon, right? So there will be no interaction, no possible interaction with, because this group is very far away from the hydroxyl group, right? So so this will be the weak binding of this molecule or neurotransmitter or drugs with the receptor, right? because this molecule containing the binding group is incorrect position, right? So this one, for example, this is mirror image of the neurotransmitter, right? So this is the enantiomer of the neurotransmitter, right? In which the hydroxyl group oriented here, but here the hydroxylated group is oriented at opposite, having the alter stereochemistry or the different enantiomer, right? So it will this molecule does not fit into this one because hydroxyl group is far away from the binding site, right? So this will be have weaker, and this enantiomer is having the weaker binding as compared to this one. So the binding, the how the weaker binding, strong binding decided where the where the binding groups are and where the what is the position of the binding groups. Size of the so these are the important. So these are the meaning that the functional group which produce the strong interaction with the receptor, right? They are the important pharmacophore of the messenger, because this helps the molecule to have the strong interaction with the receptor. Size and shape also matter. For example, here the metamethyl group, for example, this one metamethyl group, right? Here you can see in this example which we have seen in the previous one, this one it contains the methyl group, right? NH2 is only methyl group, right? But here instead of methyl there is this one methyl group, right? It has a long chain, and here also it has a metamethyl, right? And so these are the two different things, two groups which play a very important role in hardness of the binding. We hindered the binding of this molecule. So as a result of the presence of meta group, as a presence of this big hydrophobic group, this molecule have failed interaction with the binding site because of the steric hindrance produced by this method as well as this propyl group. Other design also strategies also be possible. For example, there are some extra binding region in the binding site, so which is not used by the natural chemical messenger. So the drug can be designed in this way also, by seeing that there are other possible binding sites. The drug can bind with this binding site and can act as agonist or antagonist. So there are the some agonists used in clinic are these dopamine used in the treatment of Parkinson's disease, and serotonin against use of treatment of migraine. Now designed toward the antagonist, right? So antagonist activity, so of the molecule, right? So there are two ways the antagonist acting at the binding site. Number one is antagonist acting on binding site, or the antagonist acting on out the binding site. So two ways the molecule can act uh, uh, with two ways is act with this receptor. First of all, we will see the antagonist acting on binding site is totally depend upon the structure based drug design. You can see the compound acting on antagonist at a binding site. It is a correct orientation of group which help the functional group help do the binding between the drug and the receptor, right? So that's how they bind perfectly with the this molecule perfectly bind with the receptor, right? So this is strategy based on the structural based drug design, means the structure of the receptor uh, help to form the or to synthesize the drug, or to synthesize the antagonist activity in a molecule. right? So there are several type of the pockets in the molecule in the receptor, right? So hydrophobic region, extra hydrophobic region, ionic region. So these are group can help the molecule to bind with the receptor. So meaning that this group give the idea that if the drug which have antagonist activity should be a, which can act as an antagonist should have these type of functional group: hydrophobic binding site, extra hydrophobic binding site, ionic binding site, hydrogen bonding. So for example, you can see the binding of natural neurotransmitter which act induce fit and activate the receptor, can have perfect binding site, hydrophobic site, and then this one have for the hydrogen bonding, and this one help for ionic interaction like this. It can fit into the molecule and act as a act as with this agonist. But if let's say molecule can design in a way that the some of the functionality can help to bind with the extra hydrophobic region there, so that can help to help that binding of the antagonist leading to different induced effect, right? So this will give the active induced effect. This one he gave the antagonist induced effect, because this molecule have this extra hydrophobic region which can bind strongly, and as much as the drug is bind strongly with the receptor, it can act as an antagonist. So this type of the design, the type of the drugs design, which can only be possible by seeing how the what type of the pockets receptor have, and in such a way drug can be designed. So this is how you can see the adrenergic binding site. For example, this is the binding site of the adrenergic receptor. It has there this for this portion is for hydrogen bonding. This portion is for hydrophobic interaction. This one is for hydrogen bonding. This one is for the ionic interaction. So how this compound can have like this, you can see this compound is come into the pocket, and then there are several type of the bonding: hydrogen bonding, and then ionic interaction, and this hydrogen bonding, and this is hydrophobic interaction. That's how the molecule fit into the binding adrenergic binding site. So this is the difference between the beta receptor and the alpha receptor, adrenergic receptor. So alpha adrenergic receptor does not have the extra pocket which the beta adrenergic receptor have. So if the molecule have the bulky group at this amino this at the amino part, which is the tertiary butyl group, it can fit into hydrophobic pocket of the beta adrenergic receptor, not the alpha adrenergic receptor. That's how molecule can decide whether it has to be fit into alpha adrenoreceptor or beta adrenoreceptor on the basis of the molecule. If the molecule is bulky, or if the molecule is not bulky, it can easily fit into alpha adrenoreceptor. Now the second one is the antagonist acting out with the binding site. So the antagonists which do not bind to the active site used by the this natural chemical messenger. How does it works? Right, it is work as allosteric module um, modulator, right? Some receptor have the allosteric binding site. For example, this one is allosteric binding site, this is allosteric modulator, right? So this allosteric modulator bind with this allosteric binding site and deform the binding site of the substrate, so substrate can not able to bind, or substrate cannot recognizable the binding site. This is one way of this antagonist act out with the binding site. But the second way is this antagonism by the umbrella effect. So this umbrella effect is this is antagonist. Some antagonists are thought to bind to the region, to the receptor where it feels this which are close to the normal binding site, right? So for example, they bind with the neighboring binding site, and it has some elongated portion or some extra binding site which can act as an umbrella effect, and as a result of this blocks the binding of the substrate. So this is called the umbrella